We have been engaged for over a decade with our partners at NIH in vaccine development strategies for the malaria type that kills the most, which is really pediatric falciparum malaria. And so we've been focused on that for 10 years. I think what really distinguishes this work is we began with human beings. So we obviate that whole generalizability problem. Geez, I've got this great vaccine, I can protect mice, but it doesn't translate at all to human beings. So we start with the humans. We now have serum and epidemiologic data from 1,000 children from the time of birth until 3 years of life. And we can determine who was resistant, who was not resistant to malaria at any given point in time. So the approach we took is we looked at 2-year-old children, and we picked 2-year-old children because that's the age when children first develop susceptibility, first develop resistance to parasitemia. Some of them, not all of them. And so we looked at 2-year-olds and we said, here's a group that's resistant, here's a group that's susceptible, based on parasitemia in the next year and a half. We took those resistant and susceptible sera, and we performed in this lab here what I called, DNA gymnastics. We used some fancy molecular biology to identify parasite genes that encode parasite proteins that are only recognized by antibodies in the resistant kids, but not recognized by antibodies in the susceptible kids. And it was actually my old MD PhD student, Chris Nixon, who spent about 8 weeks sifting through 2 or 3 million parasite genes to find 3 that distinguished resistant and susceptible. And that was the beginning, and that was back in 2008. Since then, the laboratory components of the project have been taken over by the first author on this paper, actually Deepak Raj, and he has been doing some extensive characterization of one of those 3 candidates. It's a protein we called schizont egress antigen 1, and the reason we named it that is schizonts are the stage of the parasite just before the red cell rupture. So remember, this parasite infects red cells, and it causes lots of the disease because your red cells pop. He called it schizont egress antigen 1 because it turns out that antibodies to this protein prevent the schizont from getting out of the red cell. We trap the parasite inside the red cell. So most vaccine candidates for malaria have tried to prevent parasites from entering red cells. We've taken a different approach. We're sort of trapping the parasite in the burning house. We moved it back into mice. We picked the most lethal rodent malaria. No one has ever been able to protect mice from this particular kind of rodent malaria. And sure enough, when we vaccinate mice with this protein, schizont egress antigen 1, we significantly decrease parasitemia and prolong survival of these mice. We went back to that cohort of 1,000 children. And instead of just looking in 10 resistant and 10 susceptible, we measured antibody responses in the entire cohort. And we asked the question, did children with antibodies to schizont egress antigen 1, or SEA1, were they protected from severe malaria? And the shocking result was children who had detectable antibodies to this antigen never got severe malaria, 0 cases. Deepak came to us after we had cloned SEA, so we had the gene in hand. But what we didn't have in the lab at that time was real expertise with in vitro culture of the parasite. And so Deepak really elevated our game in that regard, and he brought with him a particular kind of assay, a growth inhibition assay. So we had antibodies to SEA, but the question is, what do these antibodies do? And he was able to grow the parasites in culture, add antibodies, perform other kinds of manipulations to try and look at their, almost like looking at the parasite's screams of anguish as the antibodies were added to see what changes that would induce. And what he found was really quite shocking. He found that the parasites just couldn't get out of the red cell, which is, it was one of those really pretty obvious phenotypes, and so we really latched onto that. Malaria is the single greatest killer of children on the planet, single agent killer of children on the planet. And experts get around boardrooms in Geneva and debate, is it 1 million children a year, is it 1.5 million children, is it 2 million? It's like trying to rank order the infinites. Gödel couldn't solve that problem. It kills 1 child every 15 seconds. It's an unbelievable culling machine of Sub-Saharan Africa and Southeast Asia. We need desperately a vaccine for malaria. And whilst there are some things on the horizon that look promising, it is unlikely that anything that's immediately on the horizon is going to actually be sufficiently efficacious. So our next destination is an active vaccination trial in monkeys, followed by phase 1 trials in humans. We'd like to roll this out as quickly as we can.